Now, once we are done with the structure of cell membrane, let's move ahead and talk about the next important structure in the list, that is cell wall. Yes. Now, it's since so many years in our lower grades, we have been learning that cell wall is the most important component of only plant cells. But let me tell you, apart from plant cells, there are certain fungi and also some lower organisms like protists which have cell wall in their cells, around their cells rather. Now, the cell wall in case of plants has a bit different composition. It has the similar structure, it has same function of giving rigidity, protection and all these things, but the composition or the ingredients, the components inside the cell wall are different in case of plants, fungi and protists. Let's focus on those which are very important for us, that is cell wall in case of plants. Now, first thing for us to remember is cell wall is basically made up of certain ingredients. Now, these ingredients can be broadly divided in three major types or three major parts. The first one forms the matrix. Next one is microfibril. After this, the last one we have is all the other ingredients. Now, the matrix is nothing but jelly-like, that is, it will be having water. Along with that, it has other substances like hemicellulose and so on. Whereas a microfibril, as the name says, micro, small, fibril, that is fibers. So it will have fibers and hence it is made up of cellulose. And the other ingredients would include all the substances which we expect to be in the cell wall. That is, it will have waxes followed by resins. We can also have cutins, suberins, tannins, etc. All these components will together make up the cell wall. Now, let's talk about what exactly will be the structure of cell wall. Well, basically, I thought of taking this topic in a gradual flow. However, right now, I guess it is better for us to start from the point which we know and then move in the backward direction, so we do not get confused with this. Now, if we are asked that what are the different types of cells that we find in plants, we instantly answer parenchyma, collenchyma, sclerenchyma and all these xylem, phloem, all these elements. But do you know these are cells, but they are actually called vascular bundles. That means they are actually tissues that we find because they come together and perform the same function. Now, when you talk about these as a part of tissue, you need to know that they are formed from cells and these cells have certain properties that help these tissues to function together. See, basically when we say parenchyma, collenchyma, sclerenchyma, xylem and phloem and all these elements in plants, we are trying to differentiate them on the basis of two things, the function and the structure. But both these things can be differentiated when we know what are the components which are present in them. These components is what we are studying in this cell wall. Now, let us take an example. Let us try drawing the structure of cell wall to understand it better. So, for example, we consider structure of a simple cell in plants. This is how the normal structure is. So, I name this as lumen. Lumen is the cavity, but here we consider it as a cavity or a lumen because this is having all the cytoplasmic components and everything. Yes? So now, there is one layer around it and then there is a second layer around it. Lastly, we find another important layer. Which connects it with the other cells. So now, if you take a look at it very keenly, you find this orange region is the lumen. Let me take the same color to differentiate. This is the first region and same will be the second, right? Now the outermost part, let's say this region, is having different composition. And then we have this green border. So now don't get confused. Is this the structure of an entire cell? Yes. So what are these parts that we have covered? Well, this part that we have labeled as lumen is nothing but the actual cell having its all cytoplasmic components. Then these portions that we are seeing are nothing but the parts of cell wall that we are studying. So can I say this orange line around lumen is a cell membrane? Yes. And this thick region that we are finding is nothing but the cell wall. Yes. So this complete structure is representing my cell wall. That is green line, yellow line and the orange components that we have covered. Now in this structure, as we can see, there is a difference in this green color, these dotted structures and these line structures that we are drawing, right? Similarly, a cell wall is basically made up of three major components. That is this first component, this second region, the dotted lines that we find, and lastly we have this third region. Let's name all of them one by one. Let's start from the outer part. The outermost part, the green region that we find here is called middle lamella. The one immediately after this is the first one to occur, hence we call it as primary wall. And after primary it has to be, yes, the secondary wall. So these are the three major components of any kind of cell wall. So cell wall will be made up of matrix, microfibrils and other ingredients. All these structures in different components, different structures, different proportions, different ratios, they will form these three components and all these three components are making one cell wall. Clear with this? Let's move ahead and talk about them a few in details. The first component of the cell wall is called middle lamella, right? Now as the name says, middle lamella. Wait a second. We are saying this component is lying outside. So how can we name it as middle lamella? Why do we name it as middle lamella basically? Now don't get confused. As you can see here, if we notice there is another cell, plant cell over here on this side, there is another plant cell on this side. Similarly, there are plant cells around it. So it is connected with the other plant cells through this line. So this green region is nothing but the connecting link between the two cells. That is, it is a layer lying between the two different cells. Hence, it is middle lamella. So the word is middle because it is middle. It is in between the two cells. Lamella because it is one single layer. So now, this middle lamella, as we have seen, is nothing but the cementing layer. That means it is going to join the two layers adjacent in the adjacent cells, the two layers between the two cells. This is usually, now since it is cementing and it has to be like connecting link, it has to be strong. So it is made up of calcium and magnesium pectates. Pectates is having pectins. Having pectins as ingredient and having salts of calcium and magnesium for rigidity. Now this layer in case of fruits which ripe. So during fruit ripening we find this layer gets solubilized. Solubilization is getting dissolved, getting soluble. That means this layer will start disappearing, as a result of which the toughness, the thickness or the rigidity factor is lost and the two cells adjacent to each other come close and they become bulkier, and that's the reason why the fruit is ripe. It forms very soft mass, it has a tender texture. Now this was about middle lamella. The second one which we have is primary wall. Primary wall. That means the structure which is adjacent or immediately after the middle lamella. It is present adjacent to the middle lamella, so it cannot be as tough and rigid as this. It is very thin in nature. The thin nature is basically because it is hardly 0.1 to 0.3 micrometers in length. Apart from its thin nature, the most important property is that this layer is capable of expansion. This layer can expand. Now when the cell is growing, and not only growing, when it is facing Toppr pressure or osmotic pressure or any kind of pressure from the adjacent cells, it can expand, it can grow, and as a result of which it helps the cell to stop from bursting. Another important thing, now since this is capable of expansion, it is quite flexible, it will definitely be made up of microfibrils of cellulose, and it also has other components like hemicellulose and water to a very large extent. Yes, this was about the primary wall. Now the next part that we find is the most important one, called secondary wall. So basically the secondary wall, in contrast to the primary one, is not capable of growth or expansion. Why is it not? The major reason, the most important one, is that this wall is extremely thick in nature. It is approximately 3 to 10 micrometers. Yes, now it is a secondary wall structure which differentiates the various cells that we have, the various tissues like parenchyma, collenchyma, sclerenchyma. As we were going through them, these tissues or these cells are basically differentiated based on secondary wall, because the secondary wall may be absent or present in some of them. The secondary wall may also have different components, that is, the matrix that it has can have different substances, like some may have waxes, some may have suberin, some may have, suppose say, tannins or gums or etc. whatever. All these different components in the secondary wall help us to differentiate between the two tissues. So this was the basic structure of how cell wall appears. The cell wall, in a nutshell, has basically matrix, microfibril and the ingredients. Matrix is made up of hemicellulose and water. Microfibrils are made up of cellulose, and other ingredients may change depending upon the cell or depending upon whatever the location and function of the cell is. So basically the structure of cell wall appears something like this. As we can see here, there are three major layers: the middle lamella, primary wall and secondary wall. Middle lamella, not to get confused, it is not in the middle, it is to the exterior of the cell. Why is it called middle lamella? Because it is connecting the two cells in between. It's a layer, that is lamella, present between or middle of two adjacent cells, hence middle lamella. Then we have the next layer, that is primary wall, which is nothing but a very thin layer capable of expansion. And in contrast to this, we have secondary wall, which does not allow its growth or expansion, and hence it is very thick in nature. The difference in secondary wall helps us to differentiate between the cells of parenchyma, collenchyma, sclerenchyma or any other vascular bundles for that matter. Now, this was about the structure of the cell wall in general. Apart from these three layers, apart from these things, we have something else also. We have two major forms, that is, two major components which help a lot for proper functioning of the plant cell. Now the first most important component is the Plasmodesmata. I know the name may sound confusing, but it's very simple. Plasmodesmata. Now Plasmodesmata is nothing but a connecting bridge between, it's a connecting bridge between the two adjacent cells. Now you would say, we have middle lamella between the two cells. Yes, middle lamella is there in all the cells because it's an important component of the cell wall. But there are certain channels, there are certain proteins, there are certain substances apart from these which are present between the adjacent cells, and these need to be passed from one cell to another. That passing is carried out by the plasmodesmata. It's like a bridge, it's like a connecting link. So, for example, we would have one cell with cell wall, this is the cell wall of other. Now, these two sometimes will be like this, and then you find there is a gap like structure in between. Can we afford gaps between the cells in case of plants also? No. So this gap is sealed by a bridge like structure, and this bridge is nothing but the plasmodesmata. That means we will have middle lamella, and then we will have plasmodesmata in between. This helps in passing of different substances, from one cell to the other. Now, apart from this, it can also help in passing of various kinds of signals. That is, any form of message from one place to another in case of plants can be passed with the help of plasmodesmata. Now, for passing of signals or for passing of substances, we need this layer to be a bit thick. Now, this can be either simple, that means it can have a simple structure, or it can be branched. But whatever is the structure, simple or branched, usually the thickness ranges from 40 to 50 nanometers. This is the standard range in which you find plasmodesmata, the connecting links or the connecting junctions. Now, after plasmodesmata, the other important component or the other important constituent of cell wall that we need to know is pits. Now, pits are nothing but certain membrane bound or certain depressions or cavity like structures which you find, depressions, or they are cavity like structures. Now, these depressions or cavity like structures are responsible for exchange of either fluids or other substances. Now, you may wonder, we already had middle lamella for connecting link. Apart from middle lamella, we studied about plasmodesmata, which is again responsible for exchange of substances, passing substances and signal from one cell to another. Now, why do we need these pits all of a sudden all over again? Well, basically, pits are the structures which are depression or cavity like structures. These are found usually in the secondary wall. That means these are not present outside the middle lamella. These are present inside the structure of cell wall. They are present somewhere here. Now, as we can see, these green marks, these are nothing but the pits. And these pits are found where? They are found in the structure of the secondary wall. Now, why do you think there are depressions or cavities or these kind of structures present in the secondary wall? Now, just recollect, we have seen here that secondary wall is extremely thick. Now, if there are certain fluids or substances which need to pass to the cell from the cell wall, but there is no space for it, smaller substances which cannot cross the secondary wall, then in such case, it is pits which will help them. That is, those depressions, those cavities which will form structures such that substances can move inside, and from there they can go inside the cell, and vice versa also. Now, these pits which are responsible for exchange of fluids and all, these can again be either simple or they can be branched. Now, when we say simple, that means they will have a uniform width. That means all the cavities, all the depressions will start appearing same. They will just have a uniform width, and there will be these circle-like depressions or cavities. So, what exactly do we find in branched? Well, in case of branched, we will find that the pits or the cavities or depressions are surrounded by certain thickenings for purpose of support. That means, if suppose this is an example of a pit, then we find certain depositions of the cell wall components, for example, waxes, resins, cutins, suberins, any of these can form a layer around these pits, and this layer makes it branched. Thus, simple pits are those having just simple structures of cavities, and these are branched pits in which we find the pit having a layer around itself, having a covering or having surroundings of thickenings from the secondary wall. Now, this was all about the cell wall that we could study. We have seen the major ingredients which make up the cell wall, that is the matrix, the microfibrils and the other ingredients which vary from one cell to another. Then we have seen the typical structure of a cell wall. The cell wall having three major parts. Middle lamella, not to forget, it's not in between. It is present at the outside or the outermost layer of the cell wall. That is those layers which connect the two adjacent cells. Then we had primary wall, the one immediately after middle lamella, which has to be thin, it is capable of expansion and so on. Last one that we saw was secondary wall, which is nothing but the innermost thick layer, and this is the layer which helps us differentiate between the various cells of the plants. Then we saw few another important components like Plasmodesmata, which is the connecting junction, the bridge between the two cells, and the pits, which are nothing but cavities or depressions inside the secondary wall. This was about cell wall. But have you ever thought, why is cell wall important to us? I mean, what makes it so happening? What makes it so important that we need to study the cell wall so much in detail? Well, the basic functions or the basic advantages or significance of cell wall is, firstly, it's giving a proper shape or a proper structure to the plant cell. Have you ever noticed animal cell having such structure? No, because animal cells do not have cell walls. The structure of a plant cell is exclusively due to the presence of cell wall. Apart from rigidity, it is also giving the easy transport of substances, permeability of substances, as a result of which it protects the cells from attack of foreign substances. Now, cell wall is very thick, very rigid in nature. Thus, attack of microorganisms, bacteria, viruses or any organism is avoided, is hindered very greatly with the help of cell wall. This was all about cell wall. Now, after this, let's have a look at the other component, last one in the list from cell envelope, that is cell coat.